recombination between homologous chromosomes is completed by the end of. Recombination is carried out by the enzyme recombinase and it happens in Pachytene stage. So answer is option number 4. So meiosis has two stages, 1 and 2. The prophase of meiosis 1 is prophase 1 and it has 5 substages: leptotene, zygotene, pachytene, diplotene, diakinesis. The lepto word means thin chromosomes. So during prophase, the condensation of chromatin occurs. So chromatin is the entangled structure, the DNA with histone proteins arranged in the nucleus this way. So condensation will happen and slowly the chromatin will form chromosomes like this. And this pair is called homologous chromosome pair. So leptotene is when the chromatin starts to form chromosomes. So very early stage of the compaction is leptotene. So chromosomes will be seen as thin strands, very thin strands. So hence the name leptotene. Next one is zygotene. So what will happen? The chromosomes, they are arranged like this. So the other homologous chromosome will come next to it. So this kind of pairing will occur. So each have their own chromatid strands like this. So they are very close to each other. Okay. So zygotene, zygo word is for pairing. And this pairing is possible because of a complex which is known as synaptonemal complex. And the technical term for pairing is synapses. Synapses. Pairing. So pairing means the homologous chromosome, they come very close to each other, and this structure is also called tetrad or bivalent. So since the chromosomes are still in the process of compaction, this is not clearly visible. By the time the next stage comes in, they are visible. So tetrads are clearly visible in Pachytene stage. And this is the stage in which recombination occurs. Recombination. And how it occurs? By crossing over. So I will use two different colors to show you this process. So this is the chromosome, homologous chromosome. Okay. And this is the other homologous chromosome. So in this figure you can see these two chromosomes, they have exchanged their arms. So these are called the non-sister chromatid arms. This is enzyme mediated process because you have to actually break the DNA strands, the double strands of DNA, and cut out that piece and put it on the other chromosome and exchange the pieces. So the enzyme involved in this process is called recombinase. Recombinase enzyme. And this is called crossing over or recombination. And that is what is asked here. So this particular statement could have been crossing over between homologous chromosome is completed by the end of Pachytene. The next stage is diplotene. So what will happen in this case? Chiasmata is seen. Chiasmata. So the two chromosomes, they will be connected only at the site where the recombination has occurred. So they will start separating only at the site where the recombination has occurred. That part will be connected. Pairing was brought about by synaptonymal complex. So separation will be brought about by dissolution of synaptonymal complex. So in diplotin stage, you will also see dissolution of synaptonymal complex. The last one is dikinesis. This is terminalization of chiasmata. So the chromosomes will be seen as separate structures. So these are the key events of the substages in meiosis.